has been a leader in this biomarker business. In fact, I was a little bit nervous talking about that in front of her. She's a real card carrying organically finished. So.
Except those were even compounds, not odd.
Yeah. Like hold the sign. Anyway, so we're happy to have you here, and welcome.
Thank you, Peter. Well, I want to start by thanking the organizers for the opportunity to speak this morning. It's really been a lot of fun. I took a physical anthropology course as an undergraduate and did a term paper on that involved isotopes, radiometric dating, and I checked out a book, Dating Techniques in Archaeology, and I thought, oh, this could help my social life. It didn't do much for my social life, but it helped fuel an interest in isotope systems that I've had ever since.
Which doesn't help your social life.
What's that? Which has not really helped my social life either. Indeed. But I've had a lot of fun. So today I'm going to share with you some work that we've been doing in partnership between Penn State and in partnership with Gail Ashley at Rutgers, and Doris Barboni has also contributed to this project. This is with samples from lake sediments and then also soil sediments at Olduvai Gorge. And as geochemists, it's been a learning experience for us to learn about the importance of understanding hominid habitats. And I think we have some tools that we can, in our toolbox, that can help elucidate details of habitat characteristics. So, and I'd like to illustrate that with this sample set. So we're primarily geochemists. We're interested in compounds that come from plants, and the records that we study are molecular in nature, but we also couple them with isotopic signatures, both in deuterium and in carbon. And so it's the composition of the lipids tell us something about the source, the organism that made the material, sort of like the license plate. And then the isotopes tell us how that material is linked to the physical processes in the landscape, to carbon and to water. And these fossil molecules, you can think of them in similar ways that people think about other fossil archives of plant material. I think that's my pointer. So here's fossil leaves, and here's pollen and phytolith material. All of these are generated by plants. They're preserved in sedimentary environments. There's taphometric concerns. We have the same with molecules. There's source production concerns. Who makes what? How much do they make? How well is it transferred into the archive? These are all issues that are likewise interesting and important for the molecular side. Today I want to talk about a few of these characters over here, in particular the N-alkanes, which you heard a little bit about earlier today. They're waxes on the surfaces of leaves. We're going to talk, I'll show you some data for lignin-derived compounds, which is a part of woody tissues. And this is a biomarker that doesn't get as much headline, but it's one that we think is important. It tracks sedges. And so we'll be looking at woody material, sedge-derived material, and then both algal and vascular plant leaf carbon. And I'll show some data that both looks at the carbon isotopes as well as the hydrogen isotopes. So as we heard very nicely presented by Rowan Sage yesterday, and has been highlighted in a number of talks throughout the 2 days here, C4 plants are substantially enriched in carbon-13 relative to C3 plants. It's a physiological and phenomenon that drives this. The differences are quite substantial, and they're widely used. This isotopic difference is widely used to reconstruct vegetation. And often people just sort of skip that vegetation piece and go right to climate, right? They think, oh, it's dry because there's C4 plants present. And so they're very, very important indicators of climatic and ecologic conditions, that isotopic gradient. And as we've heard already, their CO2 plants are favored by low CO2, by hot and moist growing seasons. And they also have a growth extended temperature range in which they grow relative to C3 vegetation. They also release less heat as latent heat relative to C3 plants, but because they can grow in warmer seasons, they actually can have a cooling effect on landscapes. And that's been shown by work by Sharon Cowling, for example, that the extended growing season can actually have a cooling effect, despite the sort of paradox that they have less water vapor transpiration. C3 plants are not an end member, but they have a range of carbon isotopic values. This is well understood and long studied by folks such as Graham Farquhar. This is an expanded view of a leaf showing the undersurface with little pores, the stomata, that allow exchange of gas, including water, CO2, and oxygen here. And that when there's a need to regulate the gas exchange, for example, because of dryness, that influences isotopic fractionation because it becomes a transport-limited isotopic system. We see that illustrated in a very large and extensive literature on this problem or on this system. We recently published a paper where we took leaf isotopic data and calculated fractionation factors based on composition of CO2 at the time of sampling, and looked at statistically the relationship between these different isotopic signals and water availability as proxied by mean annual precipitation. And we find in this compilation a wide range of values. It's about 6 to 8 per mil across. This has been by biome, running from tropical rainforests here, which have maximum fractionation associated with a wet environment, and here down to xeric woodland or scrubland, which has a reduced fractionation and are enriched in 13C as a result of the associated with the dry conditions. This is primarily a water-driven range, but there's also differences in the community of plants in each of these biomes that plays a role in this patterning that we see as well. If we calculate based on today's CO2 values, we would see biomass ranging from -32 to -26 for our so-called C3 end member. So that range in isotopic values is something that we want to try and incorporate and think about as we reconstruct landscapes. This is work that you saw yesterday by Terry Sterling. It's a very powerful data set and very influential in our thinking about shade and other properties. You can see that the percent woody cover here on the lower axis and soil organic carbon as the vertical axis based on modern soils. And we were very excited about this and wanted to know if we could translate the soil relationship, soil organic carbon relationship, into a woody cover relationship. And you can see that that isotopic spread is much wider than what I showed you in the previous slide. The C3 end member is sort of down here in the lower third of this isotopic range, and then the extended range here is due to the mixing with C4 vegetation. So getting ourselves from a soil number to something that we measure in our lab is not actually as simple as you might think. We have to account for, fractionation during lipid synthesis, for example, and also just which molecule to pick. So if you're like me and you like to cook and you have a collection of cook tools of the trade, and you look at your assembly of spatulas and you think, which one am I going to use today? Which is the right one for this? And we do the same thing with molecules. We pick the right molecule based on what it is we want to do with it. And this particular illustration is by, it's called a heat plot that my postdoc, Anna Henderson, put together recently. It's based on the N-alkane abundance in different types of vegetation. And she put all of these data into a big compilation and then did a cluster analysis, and the composition broke out quite nicely by plant groups. And up here in the top part of this graph are the C4 vegetation. And so we can pull from this kind of analysis of modern plant data that the vegetation that derives from C3 largely is represented by alkanes that have 29 and 27 carbons. Grasses that are C4 and, are associated with C33 and 35 length carbon chains, and that 31, which is this right-hand column, actually is represented throughout. So we're using carbon 31 because it's what we consider the picking up signals from all the different kinds of vegetation that might be on the landscape. If you just wanted to pick up C4, you would look at these compounds. If you just wanted to really focus on C3 vegetation, you could look at those. So we've picked our spatula. Now we need to figure out the next part of the step, and that is to look at the isotopic difference between the soil organic matter, which is what Terry's calibration data set is based on, and the composition of those N-alkanes. And I won't go through this in detail, but let me just say that we've thought a lot about those differences between CO2, which is shown here. This is just a schematic representation of fractionation with carbon isotopes shown vertically. CO2, there's fractionation between CO2 and the leaf that differs between C4 plants and C3 plants. And then those lipids relative to leaf values are slightly different for C3 and C4 plants. And here's something that's kind of a mystery that I think is intriguing. The soil organic matter for C4 vegetation has a different diagenesis than the soil organic matter for C3 vegetation. And they're offset kind of in opposite directions. It's small, but they're in the opposite sense. So that's something that we don't quite understand, but nonetheless is shown up quite pronouncedly in the literature. And that literature compilation is shown here. And it turns out that when you look at the data for soil and for lipids, for C4 plants and C3 plants, that you get about a 9 per mil difference between the soil value and the lipid value. And it masks differences in the lipid leaf differences, and it masks differences in the leaf soil differences, but it fortuitously comes out to be about the same number. So we can apply this 9 per mil fractionation factor between soil organic matter and the lipid to Turi's data set, and that's what we've done in the next slide. So here is Turi's work down here, and the binning by ecosystem that he talked about last time, and relative to the amount of openness of the landscape, the composition of the plants that make that landscape. This is all based on Turi's publications. And then all we've done is put on top this little axis, but it's very useful for us, and that's the composition of plant waxes that we would expect given these different ecosystems. So now we have a tool that we can go and look in ancient archives and say, well, what is the landscape signal being captured in the NL canes? So the place where we've put this to work is in Olduvai Gorge, and this is work of Gail Ashley, who has done really quite elegant work on the sedimentology and stratigraphy of this basin, and has looked at the facies and the expansion and the contraction of a lake that was there throughout this time period of our study. You can see that, or maybe you can, I'm not sure in the back you can see this, but this is our stratigraphic section that shows a saline alkaline lake facies here that interleaves with wetland deposits and then an alluvial fan here on the eastern side. And so the sediments that I'm going to talk about here come from the lake deposit. She's also mapped cycles in the sedimentology that show wet and dry cyclicity on a precessional timescale. And we heard some discussion of that in the previous talk, that this precessional beat is the background beat of the landscape throughout the last 3 million years in Africa. And that shows up in these wet-dry cycles in the stratigraphy as well. So taking samples that Gail graciously allowed us to look at from the lake deposit, we looked for alkanes from plants and also from algae. And that information is shown here. So let me walk you through this. We start with the isotopic composition of total organic carbon is showed centrally here. And you can see it ranges about 10 per mil across this interval of time from about 1.9 to a little less than 1.8 million years ago. And we see this strong cyclicity that's picked up in the amount of organic carbon, which paces, and it's been shown in lakes in this region that the amount of organic carbon in lakes is preserved is greater when lake levels are higher. So this is kind of a lake depth indicator of sorts. And this beat matches each other. So we see isotopic values that track each other. Here is the aquatic plant values, and they're rather boring. They just stay kind of within about 3 per mil of each other. So algae and aquatic plants in this. Oh, that's actually C25. So these are not algae, but literal plants in the aquatic realm don't show much variation. But when we look at lipids from plants, the leaf wax is the C31. We see a very large swing throughout this interval of time. And this is about 16 per mil in range, so it's sort of the drama queen of our data set. It's showing very profound changes on the landscape from a pure, practically C4 open grassland end member to a closed woody landscape. And that beat that Peter talked about, the precessional beat, shows up in these samples. It shows up very, very strongly. We see this is now an overlay of the total organic carbon composition as well as the plant wax signal. The scales are slightly different. This is the 10 per mil scale of the total organic carbon, the 16 per mil scale of the plant waxes. And you see that this record is showing this complete transformation of the landscape back and forth, back and forth. And the timing is on a 20,000-year time scale. Here's the precessional beat that shows that. And so this is very, very consistent with what records, the records in the Mediterranean, the Sapropel records. It's consistent with the records that Gail has described from the sedimentology of the lake expansion. And it shows this transition is rather abrupt, taking place on a few thousand-year time scale between complete C4 vegetation and complete woody environment. And I think, Rick, I don't know where you are, but this would be consistent with your variability hypothesis. But what it really is showing us is vegetation on the ground. So it connects that or hopefully provides some connective insights. But that's plants. And so the question then remains, are the plants also linked to water changes? As we heard in the previous talk, those water cycle changes are thought to drive the vegetation changes. And we have an opportunity to really test that directly using the very same molecules that we measure for carbon. We can also measure for deuterium isotopes. I think there's going to be several talks on the application of deuterium isotopes as a way to track changes in precipitation composition. This is just a simple schematic of how that works. We measure the lipids from the leaves. The leaves are made by plants that get their water from their soil, and the soil is fed by precipitation. So what we measure in the leaf wax is, if we understand fractionation factors during biosynthesis and in the plant, then we can reconstruct their source water. And we will call this soil water, because we believe we're reconstructing soil water but that is more or less a proxy for rainwater. And that relationship is probably okay because plants grow when it's wet, so the soil evaporation piece is going to be minimized during those time periods. So I'm not going to go into detail of how we assign our fractionation factors for this, but anybody who's interested in the gory details, I'd be happy to talk to you later. But we simply take the observed values here, which is plotted deuterium space here. These are the measured deuterium values of our NC31. We plotted these against the carbon isotopic composition of 31 to give you that range of ecosystem from woody to grassy, back and forth. And so the whole data set that we've measured is shown here. We can then apply that fractionation factor and reconstruct the composition of rainwater, which is shown, or soil water, which is shown on the horizontal axis here. So we can translate these deuterium measurements into an estimate of rainwater or soil water. I keep saying rain because there is modern precipitation shown there, and so we're within the range of modern values. And then taking mineral archives of oxygen isotopic signatures from lake waters, which is a clay, ophthalmogenic clays. This is the work of Hay and Kaiser. And then some older work on carbonates, which gives us an oxygen isotope number. We can use our deuterium data to reconstruct environmental waters and use the mineral data to reconstruct oxygen, and then put them and compare them to modern lakes and precipitation. Modern precipitation defines a mean water line that shows here, and our soil waters, our close to rain estimates, are right on that line, and we feel good about that. Likewise, our lake waters, when we reconstruct them using the oxygen from minerals, fall on lake lines for this region. So that we feel that gives us some confidence that we're reconstructing values of deuterium that are consistent with precipitation ranges seen in that range in this area today. And it does a final thing, which is that it links that change in vegetation with changes in aridity in the environment through the changes in the composition of rain, which is known to track the amount, largely through the amount effect, which is very prevalent in this region. And so we see, as we cross from grasslands to woodlands, we see enrichment during the dry periods, or inferred dry periods from the plants, and more depletion during the wet period. So we see this consistency in hydrologic change with the vegetation change. And in the lake waters, based on algal biomarkers, we reconstruct changes in lake composition that track the source input, but they vary slightly so that those lines are not parallel, but the vertical line has a slightly steeper slope with a greater difference between them, consistent with greater evaporative loss during these dry periods and less evaporative loss during these wetter periods. So we've got a nice connection there between plants and their climate and sort of the orbital pacing of climate throughout this region. Now that's the temporal record that I wanted to share with you. I also want to illustrate how we can apply biomarkers to understand landscape changes on a spatial scale. And here's Gail, looking very chic in her field gear. And she's standing by a tufa deposit, which is a freshwater lake or freshwater carbonate deposit that has been that she's been studying in the FLK Archaeological Horizon at Olduvai Gorge. These are some of the artifacts and fossils that are found in this location. It's a very important site. It's been known for some time. I don't need to introduce it to most of you in the audience today. And it happens to correspond with one of the driest points in our record. So these very enriched values here are associated with that same type, or are associated almost spot on with this horizon. So the lake is contracted, and on what was a lake bed is now a soil horizon, and the soil is capped and based by tuffs, so there's good time control, and it's also pretty well preserved, and has fueled a lot of discussion about which I am not an expert, but I think it's important to identify that these different hypotheses for hominid behavior that come from the heterogeneous dispersal of artifacts and fossils on this landscape, how we think about the behavior that's implied by that record is testable, presumably, possibly, by the distribution of, or the type of vegetation that's present. So a time-integrated assemblage would suggest a grassland deposit. A stone cache would maybe suggest not woody, fully woody, but more bushland deposit. A central place with protection from predators that's persistent would suggest a woodland deposit. And so these are testable if we can reconstruct the vegetation on this landscape. And this is hard to do, but we're very fortunate at this locality that there's pretty good preservation of organic compounds, and we're able to make some headway here. So let's start with isotopic signatures. This is a map of the locality. This shows the incision of the gorge. There's a small fault, and the spatial scale here is about 500 meters, so half a kilometer. It's kind of a walking scale for an early hominid, perhaps. And we've measured the carbon isotopic composition of both bulk organic matter, but what I'm showing here is the leaf waxes from the vegetation itself. And we see a gradient or a change in the isotopic composition on this 500-meter spatial scale from minus 18 to as light as minus 38 or so. And you see the most depleted values are in the central samples. The northern samples are kind of intermediate, and the southern samples have the most enriched composition. And they suggest then, just based on these carbon isotope values, using the framework that we developed based on Turi's work, maybe a mixed vegetation here, C3 plants in the central site, and C4 plants as you go away. Now this is interesting, and it would be consistent with woody cover, and so that's our hypothesis we'd like to test. And as I mentioned, there's heterogeneity in the distribution of fossils and artifacts, with many of these sort of concentrated in the central sites where the most depleted values are, suggesting a woody association. So, did they have it made in the shade, Turi? The central site from the lipid isotopes was woody, and that's associated with the fossils and the artifacts. But we could test that a little further. We can look at phytoliths, and this is Doris Barboni's work, and it shows at these central sites that there's a relatively higher amount of woody plant phytoliths, a little bit of palm, and a small scattering of grass or sedge phytoliths. And likewise, we can look at woody tissues directly by using lignin material. And so here are the compounds that we use for lignin. I won't quiz you on this later, I promise. And I have a colleague, Mike Arthur, who refers to diagrams like this as chicken wire. So if that's what it looks like, you're in good company. But we like these compounds a great deal. And they tell us some really nice things. For example, these compounds here, the deMenocal compounds, come from all plants. So we can ratio to them to look for differences in the composition of source materials. And syringeal compounds come only from angiosperms. We believe this is largely an angiosperm landscape, so we shouldn't expect to see big variations in ratios of S to V. But synamel comes from non-woody tissues, so we can look for the relative abundance of these as an indicator of non-woody material relative to woody material. And that's what we will do. This is a framework for thinking about that. The S to V ratio shown vertically gives you largely a plant type difference, gymnosperms versus angiosperms. We're much more interested in this horizontal axis, which gives us synamel to venolol, it's hard to say, venolol ratio, which is a measure of woodiness. So to the right, there is less woody, and to the left is more woody. So Clay McGill, who is responsible for virtually all the data I'm showing here, has worked very hard to isolate the lignin monomers from our samples, and there they plot. So you can see here they're all nice comfortably in the angiosperm realm, so we're happy about that. And the northern sites really partitioned separately, from the central sites. And the central sites, what's of interest here, and they show very consistent values with a woody material. Well, that's cool, right? So that's kind of neat. And if we cross-plot the lipid values, which show the woodiness by their isotope signal, versus the lignin ratio, which shows us woody by its ratio, they really break apart. And we see these central sites have the low isotope values and the woody lignin character, very consistent with a woodland habitat. The southern site up here, there's just one point here, but I will call your attention to Clay's poster, which is outside this room. He's got more data up here as well. It's consistent with this. And it shows an open habitat and a non-woody plant material consistent with a grassland. And then finally, the central site, or sorry, these northern sites, where the tufa deposit is located, is suggestive of a mixed community or possibly a wetland community. And so that's something we can test a little further with these compounds. These are alkyl resorcinols, and they have a long alkyl chain, and then this aromatic ring and a couple of hydroxyl groups. And they're found in lots of plants. These are not particularly unique, except that in sedge, they tend to have a long carbon chain of about 25, a maximum of 25. They're found in many other plants, but they tend to be shorter chained. So when we find these longer chained structures, we can take that as an indicator of sedge inputs. And we find them, the sedge compounds, in the northern site. There's none that we can detect in the grassy site, and there's a hint in one sample only in the central site that we've looked at. That they're abundant and significant in the northern site, suggesting a habitat that's consistent with our hypothesis of a wetland or wet deposit, a wet region, with mixed woody and non-woody plant community. So there you have it. We have a central site that's woody, a northern site that's wet, and a southern site that's grassy. All during the very driest times, or the grassiest signal in our temporal record. So it suggests that there were sort of wet and woody regions near the lake, but the larger catchment was probably dominated by grassland. You guys hungry? Oh, you just had snack. So the last slide I'd just like to, in the spirit of the conference here, is put forward is that suggests that not only is this that these tools are useful reconstructing habitat vertically or temporally and spatially, but they can also provide us some end-member compositions that might be useful in thinking about food, dietary sources. So what's for lunch? Well, as we've heard about in some talks in the last 2 days, a number of hominids have been looked at isotopically to try and figure out what they ate. And we see a range of values. I'm not sure if this is the full range that's been observed, but what I heard in the talks yesterday was about 0 to 10 per mil, suggesting a range from C4 to C3 based diet. And we can measure the end members of these using the same compounds I've been talking about. The C3 plants, we measured about -34, and by our lipid to biomass differences, that suggests a biomass of about -26, very comfortably a C3 number. The grass lipids that are much more enriched, about -19, and again, that lipid biomass difference that we calibrate puts that at about 10 per mil, -10 per mil, which is a very enriched substrate indeed. Likewise, those sedge compounds, and I've heard it mentioned a couple of times that maybe sedge is the source of C4 carbon in some diets, the Boisei in particular, and I'm sorry, but I have to say that that's not what we see. We see a value that's -32 per mil, consistent with a biomass of -27 to -24. We don't have good constraints on the biomass to lipid differences, but Clay is working hard to get that estimate for us. So if you want to set the table with sedges instead of grasses, it's not going to explain, or at least at this site, won't explain the isotopic signals that have been represented. And then, of course, teeth and enamel can give us estimates of the protein-rich source of food in ancient diets. So with that, I would like, before I get thrown off the stage, I'll just like to wrap up with a few conclusions. We see repeated grassland forest switches in the carbon isotopes and wet-dry switches in the hydrogen isotopes. These are very strongly processionally paced, both in vegetation and in the climate in the early Pleistocene. We also, the reason I asked about the 40K world, Peter, I don't know where you are, there you are, is that we see when we do correspondence analysis of the temporal record, we see a small beat of obliquity in there as well, but it's primarily precession-based. In the FLK horizon, which is an amazing resource, we see evidence for woodland and wetland areas surrounded by grass. And the hominid fossils and the artifacts are concentrated in what we interpret as a woody site. And what's for lunch? Well, the fossil molecules give us insight into potential substrates and can help us constrain end member composition. And I think with that, I would like to thank all of you for your attention, our funding sources, and our collaborators. Many thanks.
So my
presentation, a number of questions, but the one I'll focus on here is in the FLK complex of sites. There's also a site that's equivalent stratigraphically to Zinj called FLK North Level 13, which is a less concentrated but still an archaeological site. Does that mean that there's an archaeological signal also then in one of the other habitat types that you're talking about here?
Gail, have we worked on those samples? That's
So
the northern site.
there's also an archaeological signal in the wetland area. That's interesting. Okay,
Yeah.
good. Just a further quick question, I hope. When you're looking at the habitat variability signal,
Yeah.
what do you consider to be the recruitment, the spatial recruitment of the plant waxes for that study, part of the study?
Right, so that's a really important question, is what is the temporal signal representing in terms of spatial collection? And we have two thoughts on that. One is that just plants around the margin could contribute and dominate that. And we don't see evidence for that. We can test that with the kinds of molecules that we have. And we don't see that. The other is, well, how much of the catchment was grassy or not? And the Zinj horizon allows us to test that, right? So we show, as I've just discussed, vegetation that's associated with the wetland and the woody region, but that's not what we see in the lake. What we see in the lake is the grassland. So I would infer from that the grass sort of fills the catchment, but that there's these wet spots near the lake.
Tori Sherling, I was just wondering about your biomarker for the sedge,
Yeah.
because you listed a bunch of genera, but you didn't list papyrus. Is that only because the papyrus hasn't been analyzed yet? But
I think that's correct. Do you have any papyrus? We'd be happy to analyze it.
Sure, yeah.
Okay.
Yeah, but,
Yeah, Clay is happy. Yes.
yeah. So really the question was, do you know if this compound is present in all sedges? Because papyrus is so common, it'd be really nice to know, to make sure that it's included in your
I would
sedge
love
group.
to, if you have a sample, we'd be very
Not
grateful.
in my hand, but
No?
yes.
I'm not sure if I'm allowed to cross
Definitely
state lines with papyrus. I don't know.
Definitely. You can buy it down at
Yes,
the local water
That's
garden.
a good point. I have a papyrus hanging in my hallway. Maybe I could go sample that. I don't know that my husband would like that, but. Go ahead.
In the wet cycles, because Peter showed rather extreme oscillations of wet and dry for the sapropels. Do these lakes ever expand to the point that they can spill to an outlet and then freshen, then later shrink to saline lakes?
Yeah, we have to reconstruct salinity in the lake or make an estimate of it to do the deuterium interpretation. And it never gets fresh, as far as I understand from the fish fossil record, that constrains it to more of a brackish condition. But I'm not the expert. The expert is here, and she could answer that better than I.
Well, this is a great study, so I think this is the kinds of things that we need to do to sort of link the terrestrial and the marine records. And so I sort of a question for a combination of you guys and then also Peter in terms of thinking about how can we sort of combine the results of this talk in terms of and project them to how we might be able to test some ideas that Peter presented. So if we do see a change in either the amount of rainfall and the seasonality in rainfall, and this is really like sort of in the heart of the East African rainfall zone that Peter's really talking about, if we basically had a similar degree and resolution of a record before 2, like at 2.1, right? And now this is at 1.9 to 1.8 around. Yeah. How would you expect that the change in the the record that you know, sort of the pulsing,
Right.
the beat that you're seeing, you know, could we sort of predict or you know, could you talk about how you might expect that to
Okay.
change in response to the sort of big picture changes that Peter's talking about?
Right, so it's a slice, right? We don't have that full time period, but it's in the heart of that eccentricity maximizing of the precessional range. And just at the top of a record, we're kind of coming out of that. And we see that, where the range is greater sort of in the heart of a record, and then at the top of it, it actually narrows. So we do, I suspect, and I don't have enough data to test it, but I suspect that that magnitude of switch may pace with the precessional changes that were just, I think, Rick, you showed that in your figure.
And that corresponds exactly with one of the prolonged high barrier
That's right.
that we predict.
Yeah, so this, we happened, by good fortune, to show up right in this maximum variability piece. But we see the very top of it sort of taper.
But are there sediments below that?
Oh, well, Gail?
You know, like, is there a potential to generate this record, a similarly, you know, hard-earned record below? Okay. Because I would think that these are the places where we can test these ideas, right? You know, and this is where we would need to pursue it, and it's
Hold that thought, Naomi. Let's bring it up around lunchtime. But for now, we have to continue on here. That's great. Okay. Great talk. Very inspiring. We all remember what it was like to give our first talk in front of a group of people whose papers you've been reading for your whole dissertation. So you're about to re-experience that. So Kazi Myers is a student working with Jess Tierney and myself. And we also have one other student presentation amongst this illustrious crowd. So I think it's just a great way to give someone an experience to present their work before everyone. So Kazi Myers.
Thank you, Peter. Can everybody hear me all right? Yes. Okay, great. Well, thank you to Peter and the other conveners for inviting me to give a talk on my research. So I began at Columbia about a year and a half ago, and so what I'll be presenting to you today is the research that I've completed to date for my PhD. Just to give you a quick overview about what I'll be talking about today, I'm going to first go over sort of the theoretical background for my project, which is looking at the rise of C4 vegetation over the Pleistocene and the possible climatic factors that might have played a part in that spread of C4 grasslands. I'll quickly go over the two proxies that I've developed to date from my record for Northeast Africa, trying to get a sense of what Northeast African aridity might have been doing over the Pleistocene. I'll quickly describe the basis of our project, some preliminary results. Some are very preliminary. I just finished collecting data on Monday for some of my record. Tentative conclusions I've reached so far, and then recommendations as requested by NSF. Okay, so this is the figure we've seen a few times so far today and yesterday. Looking at the human evolutionary tree combined with a couple of climatic proxies as well as vegetational and fossil changes over the last 4 million years. So as Peter really nicely introduced to you all, sapropels, these organic rich sediments deposited in the Mediterranean, which are indicative of humid cycles in northern Africa, have been observed over at least the last 8 to 9 million years. There are also a number of periods of deep lakes in eastern Africa that are suggestive of large-scale changes in hydrology over this period of time. Now, carbon isotopic records, which we've seen many times yesterday and today, suggest that there's a large spread in C4 grasslands, particularly between about 3 and about 1.5 million years ago. We can see from soil carbon isotopic records, Terry's records, that there's a large transition between these two bracketed periods, which is also, the rise of the grasslands is also strongly suggested by the increase in fossil bovids, which we also heard about more this morning. However, the climatic conditions, or the secular climatic changes associated with this increase in C4 grasslands aren't really very well understood. we know that humid-dry cycles persisted throughout the entire period, and that there were perhaps larger periods of, or increased periods of rain. However, these proxies aren't a direct signal of increasing or decreasing aridity. So so what we wanted to do in our project was to examine the climatic background to this C4 grassland expansion in Africa. So to look for signs of aridity, the obvious choice would be to first try to look at dust records in the marine basins surrounding Africa. So these two pictures here illustrate what exactly a Saharan dust storm looks like. They're extremely large in scale. You can see it swooping down on this town here. And large amounts of dust are transported off of the African continent into the Mediterranean, as well as the Indian and Atlantic Ocean basins surrounding Africa. So if we can reconstruct the dust records over the last several million years, we might think that we'd be able to get a sense of what the secular climatic changes are over time. So I've plotted up here a Sahara dust record from the Eastern Mediterranean, and I've plotted it just as the number of standard deviations from the long-term mean. So I've highlighted the two boxes outlining our C4 transition. And what you can see here is that there doesn't really appear to be a large trend in the dust over this period. You can see some orbital cyclicity, but it doesn't really appear that there are any large increases in aridity that might be associated with the spread of more open conditions and increased C4 grasses. So from that, we want to, so if this proxy isn't giving us the full picture, we can try to move on and see if there is another proxy that might give us an idea of any changes in hydrology over that period of time. So next, I wanted to just give you a brief overview of sapropels and sapropel cycles. So as Peter really nicely introduced them, sapropels are very organic, rich sediments deposited in the Mediterranean since the Miocene. So this is a nice section that shows in the little dark layers are sapropels and the light layers are carbonates. So it's actually showing orbital pacing of these sapropels, or these humid conditions in northern Africa, over a period of millions of years. Sapropels are really strongly paced by precession, or rather northern hemisphere summer insolation changes. So when northern summer hemisphere insolation was higher, you would expect to see a stronger monsoon across northern Africa, which would have led to increased freshwater runoff into the Mediterranean, and thus, through the freshwater cap and bottom water anoxia, you might expect to see increased organic matter deposition. So just as a quick analytical note, I analyze organic compounds from both sapropels and carbonates in these intervals, and the sapropels are wonderful for these biomarkers. However, the carbonates are a real devil to get anything out of. So, actually, the point I wanted to make with this slide is that there doesn't appear to be any secular changes in sapropels over our transitional period either. So we're going to move on to a third possible aridity proxy. For those who aren't familiar with it, this is an oxygen isotopic record that comes from planktonic foraminifera. These are just little marine plankton that secrete calcium carbonate shells. And we analyze the oxygen isotopic composition of the shells, which is sensitive both to changes in temperature and salinity of the waters in which they formed, as well as ice volume changes over longer periods of time. So this is a record from the Eastern Mediterranean. And you can see a general cooling associated with increased northern hemisphere glaciation over time. I've again outlined the two transitional periods. I'd just like to note that this record hasn't been corrected for increases in ice volume. So the authors looked at, or this is from Kroon. So the author is trying to look at possible precessional scale forcing of salinity in the Eastern Mediterranean over this entire time. And the results seem to suggest that there's a strong precessional peak particularly from 3 to 2 million years ago, as well as in the rest of the record. So suggesting that precessional insulation forcing is really strongly driving salinity changes in the Eastern Med. The issue with this part of the record in our transitional period is that the resolution, unfortunately, is not quite enough to capture the full precessional scale salinity changes. So in our project with Peter and Jess, we decided to actually go back to the same site and sample it at a higher resolution to see if we could fully capture the O18 changes associated with salinity. So just as the, is that an alarm? So just to, now we're getting to the actual question in the project. Was there a change in aridity from 3 to 1.7 million years ago associated with the rise of C4 grasslands in Eastern Africa? So to begin, we wanted to compare two intervals of time that had very similar eccentricity forcing, which Kate just mentioned. We wanted to remove the variable of eccentricity changes so that we could try to compare any secular changes in our proxies over time. So for our analytical approach, we used a combination of proxies to assess both Eastern Mediterranean surface water from oxygen isotopes and North African vegetation and hydrology by using compound-specific hydrogen and carbon isotopes from biomarkers in marine sediments. So we've corrected both records for ice volume changes and compared them. I won't be showing you how I did the ice volume corrections, but if anybody's interested, feel free to ask me at the end. So to do this study, we selected Eastern Mediterranean ODP Site 967, which has both good core recovery and coverage of this period of time. We looked at intervals from 1.65 to 1.9 million years ago and 2.8 to 3.1 million years ago, which I'll just go back really quickly. Today I'll be showing you data from this eccentricity interval and this eccentricity interval. And so we sampled both carbonates and sapropels and tried to analyze both proxies to capture the full precessional monsoonal forcing of our two proxies throughout the intervals of time. So I just wanted to briefly delve a little more deeply into where our hydrogen isotope record is coming from. So we sampled marine sediments from the core and extracted what we believe are leaf waxes from them. So terrestrial leaf waxes are just a protective covering on the leaf to maintain water balance within the plant. And you can see that it's on the surface of the leaf, and it helps to protect to prevent water loss when the stomata are open. So plant waxes are composed of a variety of different compounds, specifically hydrocarbon compounds, which we saw a couple of nice diagrams before. So this includes N-alkanes, N-alkanols, and fatty acids. And so very long chain or large number, carbon number compounds are diagnostic of terrestrial leaf waxes in marine sediments. So when looking at the variety of organic compounds from a sample, you try to identify those compounds that will be diagnostic of a particular plant class. So waxes are transported both as aerosols and in sediments to surrounding marine basins. So I just wanted to show you this transect of dust samples collected off Western Africa, in which the carbon isotopic composition of leaf waxes was analyzed. And it shows that the leaf wax composition is actually quite diagnostic of the provenance of leaf waxes, or what actual biomes they're coming from on land. So we're reasonably confident that leaf waxes found in the marine sediments and surrounding basins are actually telling us what's going on in the vegetation on land. So just as another quick introduction to how the delta D wax proxy works, we actually measure the delta D composition of these compounds in the waxes. However, the original signal comes from the delta D composition of precipitation. So a variety of processes affect this composition as water travels from precipitation into soil water, and then as it's transported or as it's taken up into plants and formed into waxes. So this is shown, so the overall signal that we most commonly measure is the apparent fractionation factor, the difference between the precipitation and the wax. And this is on average about -100 per mil across a variety of different biomes. So how well does this proxy actually work? Well, in the modern, a number of studies have been done to try to constrain the linearity between the hydrogen isotopic composition of leaf waxes and the composition of precipitation. And as we can see from the study here, there's actually a very strong linear relationship with a R value of 0.92 across a variety of latitudes and environments. So this is a really nice study that just came out this year by Gerson et al. And they looked at both tropical Africa and samples from across Europe. So we see that this is a very robust relationship across a variety of environments. And so we might be able to very successfully apply it in the paleo record to try to reconstruct the precipitation, hydrogen isotopic composition of precipitation further back in time. So now that we are reasonably confident that we understand this relationship, we have to first look at what the hydrogen isotopic composition of precipitation actually looks like in Africa today. So these are just a couple of maps that show you the seasonal range in hydrogen isotopes and precipitation. And you can see it's about a 100 per mil seasonal cycle change. So there are very large hydrogen isotope signals just from year to year. So on a precessional time scale, we might expect to see the penetration of the monsoon moving farther north into Africa, which would mean that plant waxes produced up here as vegetation spreads into northern Africa would start to record, hopefully on average, a more negative hydrogen isotopic composition due to hydrogen isotopes, or due to increased precipitation. Oh, I forgot to say that in tropical Africa, or in northern Africa, the hydrogen isotope composition of precipitation is very strongly controlled by the amount effect, or how much it's actually raining, which is shown very nicely here in the monsoon season and the dry season. Okay, so keeping all of these things in mind, we set up two hypotheses regarding what we expected to see in our deuterium and oxygen isotope data from the Eastern Mediterranean. So the first hypothesis was that we expect to see more depleted delta D wax and also oxygen-18 ratios during the Sapropel intervals, during which we assumed there was a stronger monsoon. And then we also expect to see more enriched values during carbonate-rich intervals. Our second hypothesis was that if the carbon isotope shift is indicative of an increase in aridity, we might expect to see the delta D wax average to be more enriched in the 1.7 million year old interval than in the 3.0 million year interval. So our hypothesis here is that we expect to see things that are consistent with an increase in aridity with more open conditions. So to begin with, I wanted to just show you what our data looked like before this carbon isotopic transition. So I've plotted eccentricity and insulation so you can keep track of what the forcing is like here. There's a slight offset, there's a slight lag in the incidence of Sapropel formation in the Mediterranean and maximum insulation. This is built into the age models. So I've shaded Sapropels here so that you can follow them with your eye. And looking first at the delta D wax results, we see that there are generally more depleted ratios in the Sapropel intervals and more depleted ratios in the carbon isotope ratios. We see about a 20 per mil range across the precessional cycle, which is about half the range of ratios observed in Eastern Africa from lake sediments that Just Tierney has developed. So this was very interesting. It appeared that our first hypothesis that this proxy would track precipitation intensity seems to be holding. For oxygen isotopes, we've observed a generally very similar trend with more depleted ratios in the Sapropels and more enriched ratios in the carbonate intervals. There appears to be some possible lags here We're not quite sure if there's an actual lead time in the oxygen isotopes relative to the hydrogen isotopes. That's something that we'll have to look a bit more into as we continue to develop this record. The ice volume correction shifts were very small during this period of time because conditions were generally much warmer. So the shifts were less than -1 per mil in the deuterium record and less than 0.01 per mil in the oxygen isotope record. So in our 1.7 million year old interval, I've set the plots up the exact same way. So in the deuterium wax record, we generally observe a very similar trend of more depleted isotopes in the sapropels and more enriched isotopes in the carbonate intervals. It's very interesting because this signal integrates a very large amount of input from northern Africa. Yet it's quite similar to what we just saw with Kate Freeman's record from terrestrial sediments that are showing a strong precessional control in the hydrogen isotope composition of the waxes. We see a slightly different relationship than in our 3-point year record in the oxygen isotopes, where it appears that oxygen is not quite as nicely tracking the sapropel changes. Again, we have to look a little bit more into this, but one thing that we need to continue to do is to constrain sea surface temperature changes over this period of time as well. So it's possible that there are some other variables that we just haven't taken into account yet. But the general trends seem to be quite striking throughout this period of time. So I just wanted to quickly look at any possible relationships between linearity between our different proxies. So in comparing delta D of wax and reflectivity, I observed roughly positive correlation between the two of them, about 0.5, which is significant at the 5% significance level. So that seemed to me that the delta D wax is fairly nicely tracking these changes, shifts from sapropels to carbonates, or from stronger monsoon to weaker monsoon. However, in comparing the oxygen 18 with the reflectivity and oxygen to delta D for both records, I couldn't really pick out any strong relationships. So now I'd like to move on to the second hypothesis, whether or not there were any secular changes or mean value changes in the two records. So the mean value of delta D wax in our younger record was -156 per mil, standard deviation of 8, and it was only about -154 per mil in the older record, which is not significantly different, and it's approximately equal to our analytical error. So this suggests to me that there is no change in the mean values of the delta D wax from 3 million years ago to 1.7 million years ago, which is very interesting, and it wasn't what we expected. So in looking at the oxygen isotopes, again, these are both ice volume corrected. We see that there is a significant difference in their means. However, we haven't yet corrected these records for sea surface temperature changes. Some of Tim Herbert's alkanone sea surface temperature work throughout the tropics suggests that there's about a 3-degree centigrade change between this period and this period of time. And so it's possible that the mean changes we're seeing are just due to sea surface temperature changes. The jury's still out on whether or not these will actually be, the means will be different once we've accounted for these other factors in the future. So just in summary, hypothesis 1, that our proxies are very roughly tracking the monsoon strength, seems to be holding. More work is needed to go into the relative timing of the variability in the delta D wax and oxygen isotope records. For hypothesis 2, we can't rule out the null hypothesis for delta D wax, that there was no change in the hydrogen isotope of wax, these wax compounds from 3 to 1.7 million years ago. So perhaps this means that there was not a very strong aridity signal over this period of time. Again, we'll have to collect more data and do more work, but for now, that's what the data says, and we can't say certainly that there was a mean change in the hydrology. For 18, we need to investigate the influence of sea surface temperatures more. So for planned future work, we are going to collect carbon isotopes from these same samples to constrain the differences in vegetation over this period of time, which we know occurred. We're going to fill in hydrogen isotope gaps. As I said before, carbonates are devilishly difficult to get organic compounds from. So we're going to be trying a couple of different methods to try to get better data from those very low concentration samples. And we also plan to analyze alkanones from our samples to construct a sea surface temperature record over this period of time as well. So to move back to this, I would say from our data, we don't see a very strong aridity signal from delta D wax of hydrogen isotopes. And so perhaps, as has been said before, this shift was more from a decrease in CO2 change. A lot more data needs to be collected to really constrain changes in hydrology. And so to get to my recommendations, I think that combining these newer organic proxies with traditional inorganic proxies will really help us get at this problem of changing hydrological conditions in Africa during these very interesting changes in vegetation as well as biology. So I also think that we need to continue to develop these proxies in the tropics and in Africa in particular. Garcin et al. Established one of the first tropical Africa studies of modern delta D wax precipitation relationships. So that's something I think should be done more, maybe in Eastern Africa as well. And I think continuing to collect and develop biomarker records from fossil hominin sites, as Kate mentioned, and as I saw in the poster outside yesterday, will be immensely important to constraining the different types of vegetation at the hominin sites. And then I think it would be very useful to compare these with existing records to try to figure out any possible climate changes that might have been going on during hominin evolution in Eastern Africa. So I'd just like to acknowledge a great number of people who have given me a lot of help and guidance in my projects and lab work and just general moral support so far. And I'd like to thank NSF for funding my graduate work as well. Thank you.
So, Cassandra, thanks for that talk. That was really interesting. So one thing I was thinking about is that even though you're not seeing these changes in, it's not certain that you're seeing changes in mean delta D and delta O18 values, perhaps the variance is actually greater, or the amplitudes between these switches might be greater. And that could be interesting because, for instance, if you see these big swings back and forth, maybe C4 grasses are responding quicker than C3 grasses after a switch, and so that's why you have this trend in over time.
I agree. That's another thing that I'm very interested in, changes in variability over time. From our existing hydrogen isotope work, we don't see significantly different ranges in the hydrogen isotopes. And so I think as we fill in the gaps a little bit more in our record, we can better test that. But the existing data suggests that there really isn't a change in the variability either, which again was not something I would have expected. Kate?
I want to congratulate you on some nice work. I look forward to following as you advance this. One of the things that we've noticed in some work that I did with Pratica some years ago now shows very clearly that the hydrogen isotope signal is biased towards when plants grow. And so the signal you're really chasing here is the growth season signal. And I'm wondering if the lack of a significant difference between the two time slices might be simply masking, or that the fact that you're only sampling the growth season is masking differences that might be more seasonally prevalent.
I agree. We also know that there were very large, and I observed in my data, there are extremely large changes in concentrations. It's actually several orders of magnitude of these long chain compounds from the sapropels to the carbonates. So we know that the vegetation was shifting, and as it got closer to the site, of course, you'd get more transport of waxes as well as more production. So I think that's an extremely valid point, and something that we need to look into a little bit more. But I think looking at some of the terrestrial records might really help to see also if there are any mean state changes. Definitely this can't exist in isolation, so combining it with different records to further understand what we're seeing will be really important in the future.
Okay. Okay, our next speaker is Sarah Feekins. Sarah was here about 6 years ago and has since gone to the other coast, and it's wonderful having you back.
Thanks very much, everybody. This is a really marvelous meeting. I really enjoy the interdisciplinary conversations. So if you think that I don't know about the thing that you work on, please grab me, don't be shy, bend my ear, and teach me about the things that I need to know. Okay, so I'm going to step back in time and think about the last 12 million years, and talking about the Marine Corps records from the Gulf of Aden. So to think about what the motivation for this is, I'm thinking about what's going on in human evolution. I mean, I personally find plant leaf waxes fascinating. Hopefully you all do too. But the extra layer is then interpreting climate, which is fascinating itself, and then the other layer is thinking about evolution. So it's triply fascinating for me, so it's really fun to work on. And so when I sent the paper into review, I tried my hand at summarizing some of the evolutionary information. I was told that I'm not allowed to wave my arms around so much, but I think that I probably can today. So here I am waving my arms. And I've summarized the molecular clock evidence from my reading of the anthropological literature, that between 7 and 5 we see the ape-human split. Between somewhere around 10 and 8 we see the gorilla-chimp-human split. In terms of the fossil record, we have the first hominoid fossil at Charora, Sahelanthropus, Aurorin, the Ardipithecus species in Australopithecus, and the emergence of bipedalism somewhere around 6 million years ago. I've also summarized the dietary evidence from Kevin Uno, who's going to talk next. Oh, and if your stomach is grumbling, we are behind, and there's 2 more talks before lunch, so be patient. So what I want to do is I want to reconstruct the environmental changes that surround these evolutionary events. That's my task. That's what I would like to do. I'm not sure if I'm going to, well, let's see, I'm not even sure if I'm going to get to whether the timings are consistent with a climate driver for evolution or whether we'll actually get to testing the hypotheses, but we'll see what we can do. Okay, so my approach to reconstruct environments is to work on marine cores that are providing a regionally integrative record of the environment in the source areas from which the dust is blowing. And I thought that that was going to be a hard one to sell because it's far from the environments in which the hominids actually lived on the ground. But fortunately, Richard thinks that it's important to work on Marine Corps records, so maybe it's not so hard to sell. Okay, and so this didn't only get the attention of the anthropological community as being something useful to work on, but also the ocean drilling program. The International Ocean Discovery Program as the next decade will be known as starting in 2013. And in the sort of white paper that they produced to motivate funding for this next decade of ocean drilling, one of the things that they featured within many of the themes that they have, one of them is Biosphere Frontiers, and Grand Challenge 7 of 14 is how sensitive are ecosystems and biodiversity to environmental change. Obviously, that's a big topic, but within that, they also focused on Peter's very popular diagram of how climate change and human evolution might interrelate. So, unbeknownst to most of you, I actually represented this community, this question, at the Ocean Drilling Program meeting in India in October. And what I was actually representing was a proposal, 724 full, which was to go back into the Gulf of Aden. The sediment core that I'm working on was drilled in 1972, before I was born. And it's a good record, but it has like something like 74% core recovery. It has lots of gaps. It's a rotary drilled core. So we'd really like new material, mostly because that core has no recovery of the upper million years. So I can't tell you anything interesting about environments over the time in which culture evolved. Unfortunately, we have a problem, and that problem is piracy. And so I learned more than I wanted to about pirates in the couple of weeks before the meeting, and This is the current map. This is the piracy attacks. Red is actual attacks, firings, boardings, and yellow is attempted attacks, and this is just in 2012. So you'll see that my hopes for taking a ship anywhere near this region are completely gone. And this is one of the most recent attacks down here. So pretty much getting anywhere near the sites of human evolution with a ship is no-go. And the Indians are actually funding this next round of the Ocean Discovery Program. And the goal is to take the ship into the Indian Ocean in 2013 and 2014 and have enough proposals to drill things. But you can see that actually this whole sector of the Arabian Sea and Indian Ocean is completely out of bounds. If Yair is still here, I want to hear about his piracy problem. Okay, so no new cores. So we have to work with what we have, which is DSDP Site 231. And the questions I want to ask is, what were regional environments like? How variable were they? And we have somewhat continuous compared to terrestrial sites, archives of regional environment that integrate, and we can capture variability. We can sample this core, we can sample it as fine as I want to scrape, but we have to think about how much time we can actually resolve in a sediment core, and we can probably realistically capture variability up to about 300 years. So this is really, really high resolution records that we can generate from these ocean cores, if we have enough time and enough money. Another question. Okay, so I just wanted to run through some of the major breakthroughs, the things that have been generated out of ocean cores, and particularly from DSDP Site 231. So these were the dust core records that I think Peter generated as part of his PhD thesis, am I right? Published in Science in '95. And these cores with the 600 numbers, these are all off of West Africa. So we're looking at more dusty to the right, less dusty to the left. So they're showing how much dust is blowing off the African continent, which can be a function of aridity and also a function of dust, of transport of windiness. And then off of East Africa, we've got DSDP Site 231, and then this site, which is off of the Arabian Peninsula that you saw with Peter's sea surface temperature records. Okay, so over here on the right is the spectral analysis of those dust records. And over here you can see the 100,000-year cycles coming in as Northern Hemisphere glaciation kicks in around here more strongly. This is the 41,000-year pacing, which again picks up with Northern Hemisphere glaciation. And this is the precessional pacing that you've heard so much about. That shows up in these dust records too. It sort of loses out to the bigger glacial cycles later on. When I look at this, the thing that I notice is that the African continent is extremely variable, and it's been variable for a really long time. It's been variable prior to the onset of Northern Hemisphere glaciation. So my title was intentionally provocative. You might not have picked up on that, but I want to point out that it's been variable in Africa for a really long time, which should be in our minds when we think about human evolution. Something else that's come out of the marine cores is that the ash layers that we find on the ground, not all of them, but some of them, make their way all the way to the Gulf of Aden. And where we have those ash layers, we have a means to date the ocean core, and we have a means to link directly to the hominin fossil. So where we have a time horizon, it's a unique time horizon, we can tie the two records together directly. And that's particularly important because we don't have perfect core recovery. Black is where I have sediment, white is where I don't. So we can't do a standard oxygen isotope chronology for these marine sediments. Something else that's come out of these marine cores is pollen. Raymond Bonfil has been working on pollen in Northeast Africa for her whole career. She's retired now, but just recently she got out her DSDP work. And so better looked at in the paper than here, but this is the tree pollen. This is the grass pollen. I cleaned up the age model so you can actually read it from where you're sitting. This is 11.5 million years ago. This is 5, and this is 1.5 million years ago. So nothing for the upper million years. Trees don't produce a lot of pollen, so that's low. Grasses are here, and this is probably the most important thing that I want you to look at. So this is about 30% grass down here. So the most important thing that I take away from this is that grasses were really important in the late Miocene. So I don't think the grasslands are a recent phenomenon in Africa. I think they've been around since at least the middle Miocene, these habitats included habitats that were remarkably open. And if you look at anything, if there's any trend at all in this data, it's sort of a decline, and then there's not really enough resolution to pick up variability in this pollen record. These are arid taxa, the Amaranthaceae taxa, that are varying dramatically in the core. And then something else that might be of interest as well, the Cyperaceae, the sedges. The sedges increase in the last maybe 6 million years relative to prior, and this might be tied in with the deep lakes in the Rift Valley. If tectonically the Rift Valley became a place with more water in it, then you could support more sedges in this part of the record. Okay, so for those of you who know me, that you know that I'm a leaf wax person, this is what I do, and so I want to focus on the leaf wax records that I've taken out of DSTP Site 231. I'm going to talk about the carbon isotope story and reconstructing the late Miocene C4 expansion, and a little bit about hydrogen isotopes and trying to reconstruct things about the wet-dry cycles in Africa, how hydrology might have changed using the complex hydrological proxy that we have in hydrogen isotopes. Okay, I am going to skip that since that was so nicely done already. Okay. So the question that I would like to answer is about the extent of grasslands in Africa. And again, to do that, I'm working in DSTP Site 231. And we already know, looking at the carbon isotope scale, that C3 is more depleted, C4 is more enriched. And we know that the N members are slightly different in the leaf waxes to soil carbonates. Something like minus 20 for C4 grasses, something like minus 30 or even more depleted for C3. C3 rainforest will be way over here. So for the last million years, this is the leaf wax data, we have a lot of variability. So the story that I want you to take away from this is that the vegetation change is variable throughout. That yes, there is a trend towards more C4, or maybe we could say more open landscapes in the last 4 million years, but remarkably, there's so much variability in any of these cycles. And if I zoomed in, you would see that these cycles make sense, not always in photogenic sort of sinusoidal curves, but they are cycles, they do make sense. So this is real variability that we can resolve if we can go in at high resolution and do enough work to generate all the data that we could generate from these cores. It's my long-term goal. Okay. So from my 2005 geology paper, just considering what we have for the timescale of human evolution, pegged by an ash layer, we can directly, the Tulibor ash layer, we can directly peg this to time and to fossils in East Africa. And we can see at this time, I've taken the scales off, but the vegetation is more C3-like, but it's incredibly variable. Around the time that Homo erectus was around, it was more C4-like, but very variable as well. So both of these species lived through incredible variability. One was more C4, more open, one was more on the C3 forested end of things. But importantly, variability was earlier than Homo. Okay, so I have been working on the Miocene. I've been going back in time, trying to reconstruct what the Miocene was like. I'm also pushing at high resolution right now, stimulated by some of the new fossil discoveries, to push back the record in this direction. So some of this is my PhD work. This represents at least a year of many people in my lab and several months on the machine by my grad student. This is my work over the past couple of years. And so this represents a huge analytical effort. And I would definitely say that it's as slow as the clumped technique, maybe even slower. So I really want to fill this in, but please be patient, I'm getting there. But I did want to put out an update to my 2005 paper and really show you what my perspective on the last 12 million years is. And so I do see a strong C4 expansion in the last 4 million years, but I wanted to see what went on before that, and importantly, how that compared to the evidence for C4 expansion back in the Miocene. And so I see C3 closed woody cover. I see a C4 expansion at around 10 million years ago. from the resolution that I have, things appear to be pretty stable all the way to about 5 million years ago. It gets pretty wooded at times through this period from 3.7 to 4.3. There are many, many cycles back and forth where it goes to values that are as depleted as back down here in the middle Miocene. And in case you're wondering what the black diamonds are, this is the age control. So these are tephra horizons that I and other people prior to me have matched up to East Africa, tuffs, and then down here we have nanofossil horizons. You can see that I have horrible age control. I have nothing between 6 and 10 million years ago, and I've tried asking for money to fix this problem, but nobody has agreed yet. Okay. So when I wanted to provide this update on what I think has been happening in terms of the leaf wax data over the last 12 million years, I wanted to also tie this into the grass pollen story that Raymond Bonfil has published. And what we see is that there's sort of a long-term decline in grass pollen. So from grasses, in grass pollen, we can't tell the difference between C3 and C4 grasses. So we know that this was a grassy place in the middle Miocene. We just don't know if it was C3 or C4. Well, the carbon isotope data can answer that question, right? And so these must have been C3 grasses, and then when C4 expanded, C4 biomass expanded, it could have been C4 shrubs, it could have been C4 grasses, but it looks like C4 grasses were coming in. So while C4 grasses were coming in, C3 grasses were going out. So that's my perspective on what must have been taking place over this timescale in the late Miocene. I also wanted to tie this onto the ground and to see what was going on in the soil carbonate records. So I invited Naomi to join me in this effort and have her synthesize and tie together all the published soil carbonate data. So this is the most up-to-date compilation. We think we've got everything. We don't think we've missed anything. If you see something missing on here, let us know. But we're pretty sure we've got everything. And so you can see that there's a huge amount of data. Us leaf wax folks are very lazy, obviously. Soil carbonates have been going on for decades, and there's lots of people's work in here, including notably Turi Serling's work from over many decades. And if we look at the data, we see the same expansion, the same trend towards more enriched values, more C4, more open landscapes in the soil carbonate record and in the leaf waxes. When we're comparing these two carbon isotope records, we need to think about what they're recording. So the leaf waxes are recording the regionally integrative vegetation that's blowing off the land surface. The soil carbonates are forming in soils over long periods of time, but they're recording a lot of spatial variability. So what you see here is the spatial variability on the ground. It's not noise, it's variability across a site. And then we can look at the long-term trend by putting that all together. This scatter is not scatter, but instead cycles of variability time that we can resolve. So they're quite different records, but quite complementary, and so we can really extract some information. Interestingly, they both confirm that this is a time which is got some more woody and C3-like values, and we see the C4 expansion here. If you're interested in the middle Awash debate, those samples plot over here. You'll note that they're very much on the C4 end of things, particularly for this period of time. So this is some of the most open and C4-rich landscapes at that time compared to other sites and other data that have been collected. Okay. One of the things that is a criticism of the carbon isotope approach to reconstructing vegetation is that the substrate is the atmospheric CO2, and that varies in time, both in concentration and in carbon isotopic composition. And one way we can keep track of that is to look at the carbon isotopic composition of benthic forams. So I pulled the data from Jim Zakos's compilation, which is the work of many, many, many paleoceanographers, and I pulled out data from sites in the North Atlantic. Because if you go to the Zakos compilation and you take the data, that includes data that jumps around from different ocean basins. And when you go back into the Miocene, there are too few records, so you wind up with jumps that are just artifacts of where records come in and go out from different ocean basins. So you can see that the carbon isotopic composition of the atmosphere as recorded by these benthic forams is reasonably constant until we get into the northern hemisphere glaciations, and then we see cycles that we could also resolve if we went in higher. So it's possible that when I generate a complete, beautiful record on the same time scale that these carbonates have been done, that I could use this to correct the leaf wax record. If there's anything, there's a general trend in this direction, so it means I'm underestimating the C4 expansion. But clearly for what I have here, that's not a factor that I need to worry about. I'd also just like to plug the next talk by Kevin Uno. His work on teeth shows that the equids are the first responders, and that corresponds to the C4 expansion in leaf waxes, but I'll leave that to Kevin. Okay, and I wanted to throw myself out there a little bit and wave my arms about CO2. I have plotted up the latest the latest CO2 reconstruction from Bierling and Royer, and this includes data from the alkanones. That's the bulk of this down here, the sort of low PCO2 reconstructions. It also includes paleosols, boron, calcium, and stomata. And so I don't place much confidence in these reconstructions because given the large error bars on many of these things, and clearly some proxies are just not doing a good job of seeing higher times of CO2, and there's a lot of proxy disagreement down in the middle Miocene. But it is interesting that the last high CO2 values that we see recorded are around 10 million years. So it's way too early for me to try to put this into the published literature. Somebody needs to do a better job of filling this in first, but it is possible that a CO2 drop around 10 million years is what drove that C4 expansion around 10 million years. Oh, and I also show the oxygen isotope curve so you can see where northern hemisphere glaciation kicks in and how that very well might be driving the C4 expansion that we see from these low values around 3.7 and then expanding onwards. Okay, so this is the latest PCO2 reconstruction from Bierling and Royer, in case you're not familiar with it. If you want to look up the CO2 data, this is where to go, and it shows you which proxies are included. This is the middle Miocene, and this is the Pliocene times of high CO2. And this all needs to be improved, and we look forward to Babel's improvements on that. Okay. So if I tie together all that data and think about what it tells me, I think that open environments have really old origins. The leaf wax and pollen together indicate that a C4 biomass increase does not necessarily mean a grassland expansion. We have extensive C3 grasslands in the middle Miocene. And I think we can start to revisit, or at least wait for that CO2 data with bated breath to see whether CO2 had a role in driving this Miocene C4 expansion. Okay. I've also been working on hydrogen isotopes. I'm not sure how well you can read this, but over on the left is hydrogen, more depleted. On the left, more enriched on the right. And this is just the Miocene from 12 to 4. And I have about 70 per mil variability in delta D. And this is plotted against the carbon isotopes. So it's also possible that this trend in the carbon isotopes might be corresponding to an aridity shift picked up by the hydrogen. I've also plotted the pollen data, the Amaranthaceae, arid taxa, and the grass taxa, which when I saw this made me worried because grasses increase at the time when I have a hydrogen isotope drop. And I've been busy playing with all of the published leaf wax literature with hydrogen isotope data. And from when we do this, we notice that there are plant type differences. And grasses, this is the fractionation relative to source water. Grasses are more negative, more depleted than the shrubs. That's the biggest difference. And so when Jess Tierney recorded a record from Lake Tanganyika that many of you will know about, the transitions were between trees and C4 grasses. So vegetation change is not something that we need to really worry about too much. At least that was the conclusion. But in this case, in the Miocene, where I see changes between C3 grasses, C4 grasses, and shrubs, I was really worried that I need to pay attention to vegetation change. So I took my delta D data and I applied a fixed correction, which is what we typically would do, to try to estimate what the delta D of precipitation would be. That's the black line. And then I also used this vegetation data and the pollen data to try to correct for vegetation change. And this depleted value becomes slightly less depleted when I apply a vegetation correction. It's not that big of an effect, but it is on the order of about up to 30 per mil. So it's something that we need to pay attention to. And I don't think this is perfect, but it's an approach that I want to explore further, where we tie together plant leaf waxes and pollen to make good sense out of the data. In grey is the modern range of precipitation in northeast Africa. So we can see that it's been dry. On the right is more dry. I would be more detailed, but I want to rush a little bit here. On the right could mean more dry. On the left could mean wetter. There are other explanations, like a stronger Indian Ocean source or a more recycled source from coming from the westerly direction. And this is work that Naomi has done with groundwater samples, which is really fantastic. The most depleted values that we see throughout the late Miocene coincide with the Messinian salinity crisis. So I put that out there for Bill, so we can talk about that more later. Okay, so bringing it back to human evolution, what I want to do is reconstruct environments, but since I'm here, I get to arm wave a little bit, so I'm going to enjoy myself. Okay, so Raymond Dart proposed the savanna hypothesis in 1925, and what the data that we have here from the pollen and from the leaf waxes is that we've had grassland since at least 11.5 million years ago. It was initially C3 and then increasingly C4. And this importantly occurs well prior to the origins of bipedalism around 6 million years ago. We can also gather some data towards the variability selection hypothesis. And we clearly see in the carbon isotope data that we've had vegetation variability since at least 4.3 million years ago. So when we think about the evolution of adaptability, we need to think about it from at least that time point. And if we look at the hydrogen isotope data, we can see that there was substantial hydroclimate variability in the late Miocene. What that means might be a little bit more harder to figure out. So what I would like to do is to continue to build high-resolution, long-term analyses needed to capture the continuous environmental context so I can answer the question about when, how much, and why variability happened, and then throw that data out there to the anthropological community to try to see how that ties in with your data. This is a huge analytical effort, and it's a hard job starting up a lab, but I have discovered a new secret, which is that there's an army of really keen people, not necessarily earth scientists, but often petroleum engineers, pre-med students who want to get some lab experience. So I've discovered that by bringing a large number of undergrads into my lab, I can start to tackle the really mammoth data set that I want to generate in order to answer these questions about climate change and human evolution. And I would like to say some of my favorite collaborators at the moment: Naomi Levin, soil carbonates, Raymond Bonfield, pollen, and Junyong Lee, who's a great isotope modeler who's really helping me to make sense of the hydrogen isotope data. So thank you very much, and I hope Kevin has some time to talk.
Okay, we have time just for one or two questions. Kathy?
Sarah, I was curious. Is this on? Yeah. I was a bit curious about pollen production by different types of plants.
Yeah.
I was wondering if you think there's any way to constrain because I know different types of plants produce different amounts of pollen. So I was wondering if you have any ideas on how to constrain the perhaps different amounts of production versus how much of each plant type is actually there.
I'm really trying to bring together the pollen and the leaf wax data, and I know that pollen production has absolutely nothing to do with leaf wax production. And so tropical trees produce a small amount of pollen, so I'm definitely underestimating that, but they fall in the mid-range of the fractionation. The things that I'm more worried about are grasses produce a lot of pollen, so maybe I'm overestimating them. The shrubs, they may also be overrepresented. So I know that it's a skewed balance that I'm playing with. In the pollen literature, people have done a lot of work on which plants produce pollen and how, catchment size considerations. In the leaf wax world, we haven't done any of that work. So what I'm doing is sort of a proof of concept at the moment, but I also have no idea source areas. Source areas might be really different for pollen to leaf waxes, and we have no data really to speak to that yet. There's one aerosol study of leaf waxes from Bermuda, a giant vacuum in the sky sucking leaf waxes, which is my best source of information.
Eric, and then we'll break for lunch.
All right, I'll make this short so we can eat. At the bottom of your the earlier part of your late Middle Miocene graph, you have around 10 shift to the right, which indicates some C4, but it seems to be much less of a shift than you get up higher. So when you talk about a change early on, am I reading that correctly, that it's relatively less than you get between 4 and 2, say?
Yeah, the most C4, like the most enriched values, definitely occur at 1.4 million years. So we do have a bigger shift to the right there. But the Miocene expansion is substantial.